taking today's class. Uh, Today's topic for discussion is measurement uncertainty and lot-to-lot acceptance testing. So let's start with measurement uncertainty. As the word defines itself, uncertainty of measurement or measurement uncertainty. What is it? First is first we have to understand what does it mean. Non-negative parameter characterizing the dispersion. हाँ रोहित नहीं मेजरमेंट अनसर्टेनिटी है. Measurement uncertainty is a non-negative parameter characterizing the dispersion of the quantity values being attributed to a measuring based on the information used. How much dispersion is there in your quantitative values is what defines the measurement uncertainty. The dispersion that is acceptable is your measurement uncertainty. Now, the ISO 15189-2022 clause 7.3.4 talks about this measurement uncertainty, evaluation of measurement uncertainty. Measurement uncertainty of measured quantity values shall be evaluated and maintained for its intended use where relevant. Standard says wherever there is a quantity value where we are going to measure in quantitative form, we should evaluate and maintain the measurement uncertainty of the that particular thing. The MU shall be compared against the performance specifications and documented. We should always compare our measurement uncertainty against our performance specifications and we should document it. There is another standard, ISO TS 20914, which provides details on these activities together with the examples. So, first thing which standard says is that the measurement uncertainty should be evaluated for all the quantitative values and it should be compared with your performance specifications which you have decided and we should document it. Now, measurement uncertainty evaluation shall be regularly reviewed. The significance of any parameter is important when it is evaluated regularly. Whether it is measurement uncertainty, it is a performance specification, it is the standard deviation, it should be regularly reviewed ki whether my uncertainty measurement is increasing or decreasing. If it is increasing, then what could be the cause for increase in the uncertainty of my result of my measurement? For examination procedures where estimation of MU is not possible or relevant, like in qualitative, you can say, the rationale for exclusion from MU estimation shall be documented. Mention it there that the measurement uncertainty will not be calculated for these parameters. When users have inquiries on MU information, another important thing which standard says that uncertainty of measurement information should always be available with the lab. You should have a list or a document which states your measurement uncertainty. If if a request comes that the lab user want to know this, the measurement uncertainty of some parameter, you should be able to tell them what is the measurement uncertainty of this test. Let's suppose I just give you an example of bilirubin. You are reporting bilirubin for a neonate and you have reported it as 23. Now, for a clinician, the bilirubin after which they do exchange is 24. In such cases, Instead of sending you a second sample and confirming your result, a lab user or clinician may ask you, Doctor, what is the uncertainty of measurement? If you know the uncertainty, my uncertainty of measurement is 0.5. Let's suppose if I am monthly checking it, I can say the maximum variation could be 23.5 or 22.5. So accordingly, they can decide after knowing the measurement uncertainty, what action they have to take. But if your measurement uncertainty is high, if you don't know it, it's difficult for us as a laboratorian to explain what is the uncertainty of measurement. When users have inquiries on measurement uncertainty, the laboratory's response shall take into account other sources of uncertainty. When users have inquiries on MU, lab response shall take into account other sources of uncertainty means you should be knowing the biological variations, the interassay variations, the total allowable errors, other sources. In the next slide, we will see what are the sources of uncertainty or what sources contribute to measurement of uncertainty. If qualitative result of an examination relies on test, which produces quantitative output data, and is specified as positive or negative based on threshold, mu in the output quantity shall be estimated using representative positive and negative samples. We should try to calculate mu where the output quantity is. You can report it as positive, negative, but if there is a threshold, we should calculate the mu. For examinations with qualitative results, which are purely qualitative, MU in intermediate measurement steps or IQC results, which produce quantitative data, should also be considered for high-risk parts of the process. MU should be taken into consideration when performing verification, validation of a method when relevant. So we should always calculate MU when we are doing a verification of a new equipment, of a new test method, or validation of a test method. In such cases, this statistical tool is very important to implement. Now, this is what I have discussed as of now is ISO 15189-2022. Next is for medical laboratories, MU estimates. What MU estimate is indicate that multiple values are possible for a given measurement. Like I have given you an example, I have reported a total bilirubin in a neonate 23. If I know my measurement of uncertainty, it could be 23.5 or 22.5, which shows that my uncertainty of measurement is 0.5. Then provide evidence that the term true value of a quantity is a theoretical concept, which also states what is the true value. I say what I have reported is the true value. It is a theoretical concept. The variation maximum dispersion in the value is the mu. Quantify the quality of result relative to its suitability for use in medical decisions. It helps us in uh, medical decisions. So the quality of a result is dependent on the MU. Assume that known medical significant bias is eliminated. So whenever you are estimating MU, bias should be eliminated while calculating it. You should, let's suppose if in IQC you are trying to measure your MU, what you can do is if there is a bias, first check with your peer group whether you are in accordance with the peer group. Then if you and your peer are have the similar bias, then it means you can define your lab mean, and after that you can observe and finalize your MU. Similarly, average of number you can see for patient results. If there is a bias, if there is a significant bias, find out the cause, remove it, and then you can calculate the MU. Assist in identifying technical steps to reduce the MU. It will help. It will also help you in reducing your MU. Allow combination with other sources of uncertainty. Can be used to determine if medically allowable analytical performance specifications can be achieved. So, MU estimate in medical laboratories can be used to determine if medically allowable analytical performance specifications can be achieved or not. This result is significant for us or not significant for us. It is medically acceptable or not. Support interpretation of patient results close to medical decision. It supports the interpretation of patient results, which are near to the clinical decision limits. Now, here we are discuss about the common sources of MU. What contributes to MU? While calculating, what we should remember. First is sample inhomogeneity. Sample inhomogeneity. Second is reconstitution procedure for lyophilized materials like calibrators and reagents. So that can contribute to variation. Uncertainty of calibrator values. We should be knowing what is the traceability of my calibrator value and what is the uncertainty of my calibrator value. This is very important. Instrumentations, electromechanical fluctuations, maintenance, part replacement. So we always say that after any major maintenance or after any major part replacement, we should do calibrations because we can nullify or we can decrease some variation or in measurement uncertainty. Then reagent and calibrator instability. If our calibrators are not stable, if we have not kept them as per manufacturer instructions, reagents and calibrators, it can contribute to measurement uncertainty. Reagent and calibrator lot to lot variability. Reagent and calibrator lot to lot variability also contribute to the measurement uncertainty. So that's why we say lot to lot acceptance testing जो होती है that वो भी acceptable range में होनी चाहिए. Otherwise, if the variation is high, it can increase the MU. Fluctuations in lab environment, personals, temperatures can also be lead to fluctuations can increase the MU. Then operator bias introduced by reading analog instrument, variable manual dexterity of manual and semi-automated methods. They all contribute to measurement uncertainty. This is the analytical part of the system which contributes to measurement uncertainty. Then measurement bias versus accepted calibration hierarchy scheme. Measurement formula, approximations, assumptions, rounding of digits, then more than one of the same measuring system for the same measurement. More than one measurement procedure for same measurement that may have different analytical performance characteristics. If you have more than one measurement procedure for the same measurement, may have different analytical performance. Of course, this we see if we have two creatinine and two instruments, even that there is a difference in the analytical performance of those. The most significant uncertainty contributions to the overall MU are captured by how you will capture the significant uncertainty contributions is long-term imprecision data obtained for IQC materials. First is long-term imprecision data. If you are reviewing your IQC material, if you are checking your LJ charts, if you are checking the monthly performance of your IQC, then if there is imprecision, and this is since long, it contributes to measurement uncertainty. Uncertainty of end user calibrator value. End user calibrator value, if your calibrator traceability is not there, if in-house calibrator, it can lead to uncertainty in your result obtainable from manufacturer or established by a laboratory that develops its own measuring system. So these are the two important things, your IQC performance and your calibrators, which contribute maximum to measurement uncertainty. What is intended to be measured? Sources of MU we have discussed, statistical estimate. Now its use, its application. Let's check it. Now, this is the application. We usually say that one standard deviation, it covers around 68.3% of the population, as when we see bell-shaped curve, and in that, while two standard deviation cover 95.5%, three standard deviation covers 99.7%. So how much is 95% covers is 1.96 standard deviation. For qualitative test, measurement is an intermediate step. Sometimes, like we have standard says, if there is a threshold, and after that we are saying positive or negative, then for that value, we should calculate the MU. If it is purely a qualitative test, then we cannot use the MU. But for qualitative tests, where measurement is intermediate, MU can be calculated and it is useful in the interpretation of the results. Then there is another clause in the ISO standard 151892022 6.5.3 which talks about the metrological traceability of measurement results. What does this clause say? Laboratory shall establish and maintain metrological traceability of its measurement results. How this can be established and maintained, the metrological traceability? They say by a documented, unbroken chain of calibrations. So it is a very important thing we say that I need a calibration certificate for my equipment yearly, whenever there is a major maintenance. Or yearly companies provide you the calibration certificate for your equipment. Each contribute to measurement uncertainty, linking them to an appropriate reference. So when they are checking the metrological traceability with their equipment, ask them, show us the traceability of their multimeters so that your calibration is appropriate. The lab shall ensure that measurement results are traceable to the highest possible order of traceability as close as possible to the international system through calibration provided by a competent laboratory. What they do in the calibration actually, they have calibration laboratories fulfilling the requirement of ISO. Where it is not possible to provide traceability, other means for providing confidence in the results shall be applied, including but not limited to the following. Result of reference measurement procedure, specified method, consensus standards. They are clearly described, accepted as providing measurement results fit for their intended use and ensured by suitable comparison. Measurement of calibration or calibrator by another procedure. For qualitative method, traceability may be demonstrated by testing of known material, previous sample sufficient to show consistent identification and when applicable intensity of the reaction. Now, so in general, what is uncertainty of measurement is uncertainty of measurement of your equipment when measurement traceability you are checking, and measurement uncertainty of your result. So measurement of your equipment is measurement uncertainty of your equipment can be maintained by unbroken chain of calibrations. How unbroken chain of calibrations? It can be maintained. What they do in that, while doing the equipment calibration, they have a reference instrument, and uh, that is called, usually they call it as multimeter, and using that they check the sensors, the voltages of your boards and everything, uh, which has a fixed temperature or a voltage. If it is beyond some defined certain limits which have been defined in the equipment during the calibration, if it crosses that, it says that adjustment is required. So always at the time of major maintenance, when they are doing calibration of your equipment, somebody should accompany the company person so that you should know what is the uncertainty of your uncertainty of your metrological system of your equipment, which contributes to uncertainty. Second thing is uncertainty of measurement is of your calibrator you should know, so that if it is high or if it is low, if it is very less, that you should know. That is very important. And uncertainty of measurement of your IQC data. Is there any imprecision? Is there any bias? If there is any systematic error, if these three things are have been covered, very your uncertainty of measurement of your analytical system of your result is appropriate. Along with that, you should know what is the biological variation for that analyte, what is the other things for that, like liable error, inter-assay precision, intra-assay precision, that all should know by the standard methods like Ricoscar. There is also, we will be going to discuss about the lot acceptance testing. So what is lot acceptance testing and why it is important? Because we have just seen in the last slide that what contributes to uncertainty measurement is lot and user calibrator lot or reagent lot to lot variation of the reagent or the calibrator. So whenever we are receiving the reagents, we should uh, receive the reagents or of preferably for a little longer time, like 2 months, 3 months. Our lot should not be frequently changed. But if, let's suppose, our lots are frequently changing, or even if we are receiving after some lot is changed, what we should do? We should know what is what is my IQC value on the previous lot and what is my IQC value on the current. Along with that, if possible, we should run 2 or 3 patient samples and we should see what I have reported on the last lot and what I have reported on the new lot now. Of course, there will be a difference. Difference will always be there. So that difference should be acceptable. That acceptable criteria you can define for your lab. Usually, if you follow the guidelines of some accreditation body, like I give you an example, if you are following the guidelines of NABL, you are trying for NABL accreditation, they have said that lot to lot variation, the maximum acceptable variation should be 10% or it should be less than 10%. So this is how you can define the, what will be the maximum variation I will accept for my parameters. So that is lot to lot acceptance. If the variation is low or less, it means there will be uncertainty of your result is less. It's measuring, it's more comparable with your previous values. And if the difference is high, so it will increase the uncertainty of measurement for that analyte. So usually in lot acceptance testing, how we record it as, you can record it in the form of a format, or you can also record it where you are managing your daily inventory. So write down the date and the parameter, QC level 1 or level 2, or patient ID. Define it. You will be checking the lot acceptance by patient value or by QC result. Then what was your old lot value achieved and lot number, and what is your new kit lot number and value. And then enter the result, calculate the difference, and if it is acceptable, accept it. If it is on a higher side, check it with another level or with the patient sample, keep it under observation, check your calibration, lot number and instability, because they all contribute to the uncertainty of your result. This is the format for lot acceptance. Thank you. This is all about lot acceptance.